All right, today we're diving into a really fascinating and, frankly, pretty enigmatic anaerobe, Fusobacterium naviforme. Now, this whole explainer is built on a comprehensive review we pulled together using Gemini Deep Search. For all of you who love to get into the weeds, you can find a full list of all the primary sources in the notebook document that goes along with this.
Okay, so here's our game plan. We're going to take that classic bench-to-bedside approach. We'll kick things off with the big question. What even is this organism? From there, we'll walk through how to actually identify it in the lab, what it means for the patient, and we'll wrap up with how we, in the lab, can help ensure diagnostic quality and even prevent infections. So first things first, let's set the stage. We need to understand the clinical context for Fusobacterium naviforme. You know, this is one of those bugs that lives with us quite peacefully, right up until it gets the perfect opportunity to cause some real trouble. Now, on paper, Fusobacterium naviforme seems pretty straightforward, right? It's a gram-negative, anaerobe, a normal resident of our mucosal surfaces. Infections are almost always endogenous, meaning they come from our own flora when something breaks down a barrier. But what's really interesting is how it kind of lives in the shadow of its more infamous cousins, like Fusobacterium nucleatum and Fusobacterium necrophorum. And because of that, it's just far, far less understood. And this lack of characterization leads us straight to the single biggest challenge with Fusobacterium naviforme. For decades, we haven't been entirely sure what we're identifying, and it's all thanks to its incredibly confusing and unstable taxonomy. Believe it or not, this organism's identity crisis has been going on for over a century. It started out as Bacillus naviformis way back in 1909, got its current name in 1970, but the real bombshell, the one that changed everything, dropped in 1991. And that discovery eventually led to a proposal just this year to move it into a completely new genus. And here it is. This is the data that threw a massive wrench in the works. Back in '91, Garbia and Shaw showed that the clinical isolates we were calling Fusobacterium naviforme, based on their appearance, were, genetically, from a different planet than the official type strain. I mean, just look at these numbers. The G plus C content is off by nearly 20%. There's almost zero DNA homology. We are not just talking about different species here. This is evidence of entirely different genera. So, this massive genetic difference, now confirmed with all our modern whole genome sequencing tools, is what's behind the 2024 proposal to rename it Zandiella naviformis. And look, this isn't just some academic debate. This has very real, very practical consequences for every single one of us working in a diagnostic lab. So, this brings up the million-dollar question for the modern lab, right? If our MALDI-TOF databases, our trusty workhorse, are built using the spectral fingerprint of that original type strain, can we even trust it to correctly identify these genetically different clinical isolates that we're actually seeing in our patients? It is a huge, huge potential pitfall for misidentification. All right, this brings us squarely to the lab bench. With all this complexity swirling around, how do we reliably identify this organism when it finally pops up in a culture? Well, the basics still give us our first clues. On a gram stain, you're looking for those classic spindle-shaped or fusiform gram-negative rods. And as you'd expect from an obligate anaerobe, it's pretty darn fastidious. It needs enriched media and really strict anaerobic conditions to grow. And even then, it produces these small translucent colonies after a day or two. Its biochemical fingerprint and resistance profile also help narrow things down. It's indole positive, and it kicks out butyric acid. And, critically, all fusobacteria are intrinsically resistant to aminoglycosides, which makes sense, because that drug uptake mechanism needs oxygen, and these guys hate oxygen. We also fully expect to see high MICs to macrolides and fluoroquinolones. But, and this is a big but, given the whole taxonomic mess we just talked about, we simply cannot stop at a presumptive ID. After you get a MALDI hit, which you have to take with a grain of salt, the definitive answer has got to come from molecular methods. 16S rRNA gene sequencing isn't just for research projects here, it's the gold standard, and you could argue it should be the standard of care to be absolutely sure of what you're dealing with. Okay, let's move from the bench to the bedside. Once we have a solid, accurate identification, how does that information translate into actually managing the patient's infection? First off, there's a really critical point we need to understand about its pathogenesis. Fusobacterium naviforme is missing something called the FAD-A adhesin. Now, this is a key virulence factor that its cousin, Fusobacterium nucleatum, uses to latch on to host cells. This is a huge distinction. It tells us that Fusobacterium naviforme must have a completely different playbook for colonizing tissues and causing disease. Because this bug is often part of a polymicrobial infection, treatment is all about aggressive source control and really broad anaerobic coverage. This table here compares some regimens from the Dutch Working Party on Antibiotic Policy, or SWAB, and the IDSA here in the States. You can see the themes are pretty consistent. Beta-lactam/beta-lactamase inhibitors, carbapenems, and metronidazole are always the main players.
Now, in pediatric cases, source control is just as critical, maybe even more so. The antibiotic therapy really hinges on careful weight-based dosing. So here on the slide, you can see some of the typical dosing schedules for the core antibiotics we use to treat these kinds of infections in kids.
All right, now we come to one of the biggest practical headaches for any clinical lab. How do we actually perform and, maybe more importantly, how do we interpret antimicrobial susceptibility testing for this organism? And here is where we hit a massive data gap. The European Committee on Antimicrobial Susceptibility Testing, EUCAST, they haven't published specific clinical breakpoints for a Fusobacterium naviforme. What that means for us is, we can get an MIC value from our machine, but we have no official standard, no ruler to tell us if that number means susceptible or resistant. But hey, all is not lost. EUCAST actually provides some really clear tiered guidance for exactly this kind of situation. The first and best option is to find a closely related species that does have breakpoints and use those as a stand-in. If that's not possible, we can fall back on other tools like epidemiological cutoffs or pharmacokinetic data. So the most practical solution here is to use the established EUCAST breakpoints for Fusobacterium necroform as a proxy. But the key, and this is absolutely non-negotiable, is to include a very clear disclaimer in the final lab report. You have to state that the interpretation is an extrapolation based on a related species.
Okay, let's wrap this all up. Let's tie it all together by looking at the role of the microbiologist in infection prevention and in this broader concept of diagnostic stewardship.
Because these are endogenous infections, we aren't really talking about patient-to-patient transmission. Our biggest impact on infection control is actually through diagnostic stewardship. It's about making sure that the diagnosis is correct from the very beginning. And that starts with a perfect pre-analytical phase. It means we have to advocate for high-quality specimens, aspirates or tissues, not swabs, and make sure clinicians are using proper anaerobic collection and transport systems to keep these oxygen-hating bugs alive on their trip to our lab.
So this whole journey leaves us with a really important question for the future, doesn't it? For organisms like Fusobacterium naviforme, where its very definition is shifting right under our feet, how do we, as a diagnostic community, make sure our methods, our databases, and our interpretations evolve right along with the science?
Whew! We hope this deep dive into the weird world of Fusobacterium naviforme has been useful. How does your lab handle these kinds of taxonomic challenges? I'd love to hear about it, so let us know in the comments below. If you found this explainer valuable, please subscribe for more. And don't forget, that full list of sources is waiting for you in the accompanying notebook document. Thanks for watching.